In this video, I will explain how to fabricate microfluidic devices. A microfluidic device is a centimeter square chip with grooves with a width and depth of less than 1 millimeter that allow fluid to flow through them. Also called lab-on-a-chip or biochip, there are various types depending on the purpose. Chemistry, biochemistry, cell manipulation and analysis can be carried out efficiently on a small scale. Here, I explain how to make silicone rubber microchannels used in the lab. Silicone rubber is officially called polydimethylsiloxane, PDMS. Commercial microchannels are often made of plastics, but PDMS is used in research and development because it can be molded at low temperatures and is easy to bond. First, draw the design of the microchannel using CAD. It's hard to know what kind of design to draw from a scratch, but it's a good idea to start by finding something in papers similar to your purpose and copying it. Next, the design is transferred to the photomask after data conversion. A photomask is a glass plate coated with a thin chromium layer and a photosensitive photoresist. A laser or ultraviolet light is then applied to draw the design on the micrometer resolution. Then, the chromium layer is partially removed by etching, and a photomask with transparent flow channel parts is completed. Normally, the apparatus to make fine photomasks is very expensive, so if you don't have an access to it, you can outsource it to a company. Next, fabricate a photoresist mold. Microchannels are often relatively deep, with grooves ranging from 10 to 100 microns depth. Thus, thick negative photoresist is used. First, liquid photoresist is applied onto a silicon wafer using spin coating. The spin speed and photoresist viscosity determine the thickness. Next, bake it on a hot plate or oven to evaporate the solvent. After that, we layer the photomask we made earlier and irradiate it with ultraviolet light. Since UV light only passes through the transparent parts of the photomask, the photoresist will only harden in the areas that are exposed to UV light. After that, a post-exposure bake is performed to further facilitate polymerization. Finally, when the photoresist is immersed in a developer, the uncured photoresist dissolves, leaving only the hardened portion.
This creates a three-dimensional structure on the surface of the silicon wafer.
Next, transfer this to silicone rubber. The method is simple. First, a silicon wafer with a mold structure is placed in a container. Then, silicone rubber mixed with a curing agent is poured in and degassed to remove air bubbles. At a raised temperature, it will cure in about 2 hours. Finally, by cutting it out with a knife and peeling off the cured silicone rubber, you get a structure with the grooves that serve as flow channels transferred to it. In order to pump the solution through the channels must be closed by bonding to another plate. First, use a punch to open holes for the fluid inlet and outlet. The PDMS surface is then activated by irradiation with oxygen plasma. In this state, attach it to a glass plate or another silicone rubber plate and heat it for a while to bond the plates together. This process produces a very strong covalent bond. Finally, connect the tube to the pre-opened holes and pump the liquid using a syringe pump or pressure-controlled pump. Using this method, we create various types of microfluidic devices. For example, this channel is used to generate droplets, which are uniform water in oil droplets. They are used as very small volume test tubes with a massive number. It is also possible to separate each cell one by one in these droplets and see the properties and functions of each cell. This channel is used to create capsules with an oil shell layer to create liposomes. This is called a microchamber array, and is used to confine cells one by one and examine their properties. This channel is called the deterministic lateral displacement, or DLD, which separates cells etc. by size. We also fabricate various types of microfluidic devices and use them in experiments. Thank you for watching. We would be happy if we could contribute to society with microfluidic devices.